Hello, everyone, and welcome to GenScript's 2020 Spring Webinar Series. My name is Grace Tan, and I am the Product Manager for Reagent Antibody Services at GenScript. I have the pleasure of moderating today's webinar. If you have a question for the speaker or the moderators, please type it in the chat box for us to see. The speaker will be taking your questions at the end of the webinar. However, if we run out of time and he is unable to get to your question, we will be sure to send his response to you via email. So without further ado, let me introduce our speaker today, Dr. Joshua Burgess. Dr. Burgess, Advanced Queensland Early Career Research Fellow at Queensland University of Technology, where he also received his PhD. His research within the Cancer and Aging Research Program at QUT is centered on determining how genome instability contributes towards the development of cancer, and his research accomplishments have laid the foundation for the development of a companion diagnostic for cancer patients undergoing PARP inhibitor therapy. Today, Dr. Burgess will be providing us with an introduction to DNA damage pathways, updating us on the use of PARP inhibitors in cancer treatment, as well as sharing more about his work on the characterization and development of a novel protein biomarker for cancer therapy. Thank you, Joshua, for joining us today. Please take it away.
Cool. Thank you for the introduction. As mentioned, I'm a member of the Cancer and Aging Research Program at the Queensland University of Technology. So today's seminar is titled Identification and Characterization of a Novel Biomarker for Cancer Therapy. So today's webinar has three main learning points. So firstly, I'll introduce the DNA repair pathways. Secondly, provide an update of DNA damaging PARP inhibitors. And third, highlight the value of biomarkers. In addition to this, I'll highlight some of my work I've been undertaking in this field of research. So before getting into the research, I actually just wanted to highlight my research team, in particular CARP management team. So here you'll see it's quite multidisciplinary. So we have Kenneth O'Byrne as the clinical director of our team, where he really helps progress the basic research we do and taking it into the clinic, as well as we have Derek Richard being the scientific director, which focuses on the basic research we undertake in the lab. And finally, Rosany Kozak, which is the CEO who directs the commercialization connections of our projects. Now, our lab actually has over 50 researchers and students in our team. So our lab really has a core focus on DNA damage repair, and this really provides a foundation for all our research projects. So this feeds into cancer work. So our team is actually progressing to a phase 1 clinical trial on our first cancer therapy. As well as this, we develop new biomarkers, and with the main focus of this cancer area is to really improve patient survival. The DNA repair studies has also taken us to look into the aging field. So our real goal is to improve the healthy lifespan and reduce aging-related diseases. This is things such as Alzheimer's, and we're doing this through the development of new novel compounds. So as I mentioned, the core of our lab is DNA repair pathways. So there's a multitude types of DNA damage events that occur every day in our cells, and this actually requires a number of different repair pathways. So here we can see bulky adducts, which are repaired through the nucleotide excision repair pathway. We also have base mismatches, which are repaired through the mismatch repair pathway. Double-strand breaks, which are defined as the most deleterious type of DNA damage, which can be repaired through either non-homologous end joining or homologous recombination. And finally, single-strand breaks, which are repaired primarily through short base excision repair. So our lab actually has a primary focus on double-strand DNA break repair. So you can see here through homologous recombination, the strands are initially bound by mRNA complex, and this allows the resection of the DNA. So this results in single-strand DNA fragments, which are bound by Rad51. And this Rad51 binding facilitates strand invasion, where a homologous DNA sequence is actually able to be used for the repair of the DNA. Due to the necessity of the homologous template, this pathway can only actually function within the G2 and M phase of the cell cycle. The alternative pathway, being non-homologous end joining, is a bit more simplistic, which is the recruitment of DNA repair proteins and simply the religation of the broken ends. This actually has a high risk of loss of genetic information. So you'll see a similar stepwise process in the other DNA repair pathways, with basic steps of DNA recognition, followed the recruitment of additional DNA repair proteins, and then the excision of the damage, and finally the synthesis and ligation. So knowing all this, I have this slide just to show that we actually target the DNA with a number of cancer therapies. So here you can see platinum-based DNA damaging agent, which causes DNA cross-links, as well as you have topoisomerase inhibitors, which induce DNA adducts, as well as double-strand breaks, and of course radiation causing double-strand DNA breaks. There are some issues with these sort of treatments though, as they often have a lot of side effects, and this is really detrimental to the patient's health. So really moving into more targeted therapies is the goal. So one of these targeted therapies is really been through synthetic lethality. So here I'm just going to discuss the example of PARP inhibitors. So following a DNA damage event, it can be repaired through homologous recombination. So this is dependent on some genes you may know about, so the breast cancer associated genes or BRCA genes, and this results in the effective DNA repair. But actually in cancer, a lot of the time these BRCA genes are mutated, so the cells actually rely on alternative pathways. So this alternative pathway is dependent on PARP. So if we inhibit PARP using a PARP inhibitor, so here indicating olaparib, this actually results in the DNA being unable to repair specifically in these cancer cell lines. So you can kill the cancer cell lines whilst the normal tissues have minimal effect. So this can generally be better than the chemotherapy. So how do PARP inhibitors actually work? So it's actually had some sort of contradictory mechanisms. So I'll go through some of these briefly now, what people are proposing, but you can see in this scan here that they can be quite effective. So being the tumor being like undetectable following PARP inhibitor treatment in one of these ovarian cancer patients. So the first mechanism that has been proposed is really the single strand break repair being inhibited through PARP inhibitors. So you can see here, PARP inhibitors are actually required for the parylation of proteins for the successful repair of lesions. However, studies have identified that if you actually inhibit cells with PARP inhibitors, there's not actually a massive increase in single strand breaks. So this doesn't really fit well with this mechanism, as well as this depletion of PARP through siRNA hasn't shown the same toxicity as the inhibitor itself. So this really suggests potentially additional mechanisms. So an alternative explanation has been replication forks. So stalled replication forks can occur, and PARP proteins actually been shown to be required for the restart of these replication forks. So in the absence of PARP1 or the inhibition, there seem to be replication fork collapses, and this actually requires the repair of homologous recombination to fix this damage. And as I mentioned, in the BRCA deficient cells, which have been described as sensitive, they have defective homologous recombination, so they result in cell death. Additional to this, PARP trapping has actually been a focus for a number of PARP inhibitors, with the goal of these inhibitors to make PARP be stuck on the DNA. So this really creates an obstacle during replication, so that when the replication happens, the replication fork hits this bound PARP, and this results in a double-strand break, once again requiring homologous recombination for the repair. There is some data to support this theory, however, it hasn't really translated into clinical findings, with the best PARP trapping inhibitors not actually resulting in the best clinical benefit. Due to PARP's role in replication through its parylation of proteins for the activation and deactivation, it has been proposed that without this mechanism, the replication speed isn't controlled properly. So this results in actually faster replication. So this can lead to more single-strand breaks. This is another proposed mechanism. So the increase in single-strand breaks, increasing apoptosis. PARP inhibitors have also been shown to alter the transcription of proteins. PARP1 in particular is associated with the activation of E2F1, which is a transcription factor of actually a number of DNA repair genes. As well as this, some screening studies identified factors including p53 and NF-kappa B being altered by PARP inhibition. So this will result in further defects in the DNA repair machinery. The final proposed mechanism is actually just the increase of non-homologous end joining. So PARP inhibitors have been shown to increase the phosphorylation of DNA PK substrate, which consequently increases non-homologous end joining. So in combination with the loss of HR in the BRCA mutant cells, for example, this would force cells to use the slightly more error-prone mechanism of non-homologous end joining. So over time, this would result in the accumulation of DNA damage and the subsequent cell death. However, from all this data people have been generating, it's been quite clear that the proposed mechanisms don't seem to be one in particular, with more likely that multiple mechanisms lead to toxicity seen from the PARP inhibitors. Here I'm just looking at the current status of the PARP inhibitors at market. So 2 have been approved for the treatment of ovarian cancer with BRCA mutations, being olaparib and ruciparib. whilst talazoparib being approved for breast cancer, in addition to niraparib being approved for the treatment of homologous recombination deficient, or HDR, ovarian cancer. So one major caveat for this treatment option is actually the requirement for an accurate biomarker, so currently being the BRCA or HDR status selection, but this is only actually resulting in approximately 30% response rate in patients. And clearly there's a massive financial market for this, as well as a consistent growth in this market. So the development of a better biomarker has really been a big focus of my study. So what really makes a biomarker? So biomarkers can be basically anything from RNA, DNA, as well as protein levels, or even the mutation status of these. So it can be tested within the tumor, as well as the normal tumor, normal tissue, based on which marker it is. So the use of the biomarker can apply additional information to gain on the cancer case, which can assist in things including the patient diagnosis, the risks associated with different treatments, as well as providing therapy selection guidance, and simply the patient prognosis. So going through some examples of biomarkers which are currently in use. So this for diagnostic biomarkers can include things such as genomic classifiers, so sequencing the genome to identify the likely source of the tumor. So serum markers including osteopoietin in ovarian cancer, as well as being prognostic markers. So you have PSA levels for prostate cancer, which indicate the rate of tumor relapse, as well as PTEN mutations, which suggest a poor patient outcome. As well, there's a large array of single gene prognostic factors. So predictive markers as well, such as PD-L1, which indicates the likelihood of response to immunotherapies. TRK fusions is another predictive marker for treatment, as well as, of course, the previously mentioned BRCA for PARP inhibitors, and EGFR and ALK mutations for the treatment of their respective inhibitors. So the development process of biomarkers can be quite varied based on the biomarker. Now, there's a few basic steps which would be characteristic of all biomarkers. So initially, it'd be the discovery phase, which would be the basic research, as well as it could be through targeted screening techniques or even bioinformatics analysis. So during this phase of detection, the system needs to be optimized, and then following this, further validation will be required. So depending on the biomarker, this can be done through cell line work. PDX samples could also be utilized at this stage. So really the goal of the validation stage is to provide some foundation to support the efficacy of a clinical trial. So for clinical trials, initially it'd be great to use a retrospective study if it can be utilized. So this is the main goal of this is to validate the biomarker and show that it's actually better than the current clinical practice. And if all this is successful, then we can move into the approval phases. So as previously mentioned, the PARP inhibitors currently use the BRCA and HDR status as their biomarkers, with BRCA being the mutation status of the BRCA gene, and HDR can actually be measured through a few different methods I've indicated on the slide. But outlined here, the testing for the biomarkers can be from a number of sources. So you have germline mutation testing as well as tumor testing. But primarily with the germline testing for the BRCA, it actually indicates a greater risk of breast cancer, being from a 12% up to a 60%, as well as ovarian cancer from 1.4 to 15 to 40% chance. So what is the benefits of PARP inhibitors? So far, they've actually been quite promising with, as I mentioned, approximately 30% of BRCA mutant patients responding to treatment. This statistic has been quite varied, with up to 50% response in some studies, whilst as low as 20 in others. And this has been primarily based off their criteria of where they cut off their response. Looking at some studies in particular, so this breast cancer study demonstrated an increase in survival from 4.3 to 11.2 months in the BRCA mutant cohort, whilst the BRCA wild type only had a 2-month improved survival. Similarly, can be seen in an ovarian cancer study from 13.9 to 49.9 months survival in the BRCA mutant, while still a great increase in the BRCA wild type from 5.5 to 19.1 months. So this can be seen that there's actually a good proportion of patients who are actually responding in this BRCA wild type tumors. And some studies in particular with niraparib and reciparib have indicated that patients outside this BRCA mutant cohort could have a good response and they should be treated. This is just a Kaplan-Meier plot representing one of the studies. We can see there's actually quite a good benefit where you have the olaparib treated cohort having significantly longer progression-free survival compared to the placebo-treated cohort. So moving into my research now. So really, I'm trying to develop a new PARP inhibitor biomarker, and I'll go through some of the data that I've generated to date. And the need for the new marker is basically because only 2% of breast cancer and 10 to 15% of ovarian tumors present with this BRCA status. And with only the 30% of people responding to this cohort, it's clear from these studies that agents can be beneficial beyond this small niche of patients, and to become accessible to a wider cohort of patients, really a new biomarker need to become available. So the product that we've been developing is actually a monoclonal antibody against our identified biomarker. So to secure this development, we've actually got a patent on the target, as well as up to now, we've tested this marker in a large array of cell lines, as well as PDX models. We're currently working on retrospective clinical trials. And it's also important to have the resources available in the lab, as well as we collaborating a lot on this project to ensure that we can actually successfully complete this project. So how do we discover our biomarker? We're working on the fundamentals of DNA repair. We identified this novel protein through a correlation screen with one of the main proteins we worked on in our lab. So initially, I wanted to assess if this new protein had any role in DNA damage repair. So this is a neutral comet assay. So you can see here, the cells are basically suspended in agarose and a current run across them. And the longer the tail indicates more fragmented DNA. And if I simply deplete the cells of this new biomarker, indicated here with KD for knockdown, you can see a significant increase in the length of a comet tail, indicating more DNA fragmentation. So the cells are basically not stable and accumulating DNA damage events. Next, I really wanted to see, are they required for the repair of DNA damage? So here I've used gamma H2X foci, so using immunofluorescence. I've once again looked at the cells following depletion of the biomarker, indicated with a KD, and following induction of DNA damage using ionizing radiation, there's a massive increase in gamma H2X foci, and over time they're being repaired. However, in the depleted cells, there's actually a delay in this repair, indicating this protein may actually be required for the repair of DNA damage. The next assay I wanted to show is the homologous recombination assay. So this assay is a GFP reporter system where there's basically a plasmid integrated into the genome, which reports back with a GFP fluorescence if homologous recombination has occurred successfully. So once again, depleting the cells of biomarker, we can see a significant decrease in homologous recombination. So this really leads to say that the cells require this protein for effective DNA repair. So based off these findings and some others, we were proposing that this protein may be a good biomarker for PARP inhibitors based off the synthetic lethality I discussed earlier. So to investigate this, I utilized panels of breast, lung, and ovarian cell lines. So here you can see each one of these points on each graph represents one cell line. And I've calculated both the expression of the protein as well as the IC50 to the PARP inhibitor. And we can see across the different PARP inhibitors, so we have olaparib, talazoparib, and vilaparib I've done for breast cancer. You can see a strong correlation with the low expression of the protein resulting in low IC50, so a greater sensitivity to PARP inhibitors. This could also be seen within lung cancer and ovarian cancer. So to do some validation on these findings, I depleted the protein again within the cell line panels, and once again calculated the IC50 to olaparib. It was hypothesized that depletion of the protein would increase sensitivity to the PARP inhibitor, and in both the breast and lung cancer cell panels, we can see that the depleted cells had a lower IC50, so became more sensitive as we predicted. In addition to this, I was also able to stably overexpress the protein in cell lines. And we can see when we overexpress the cell line over the dose range, they become more resistant to PARP inhibitors. So it's all really fitting together quite nicely, but this is clearly all just in cell line models, so we need to go further than this to validate it. So to do this, I first looked at some PDX models. So here we can see I got a panel of patient-derived xenografts, looked at the expression of the biomarker across them. So we can see we had some high and low expression PDXs. I did this both through Western blot here and also did IHC scoring, as this is what would primarily be done within a clinical diagnostic application. From this, I was able to select one low PDX and then check to see how it would respond to a PARP inhibitor. So here I treated with olaparib, and we can see in both a low dose and a high dose olaparib really had a great response to the PARP inhibitor. Whilst alternatively, the high expressing PDX, which was selected, had quite a poor response to PARP inhibitors. So here you can see that it only just met statistical significance at the highest dose of olaparib. Additional to this, I was actually able to have a look at a retrospective PDX panel with known reciprocity sensitivity. So for this study, it's important to say that we're actually blinded to the sensitivity of the samples until our expression was determined. And this ended up being consistent with our previous findings. As you can see, the sensitive cohort had lower expression compared to the non-sensitive cohort. In addition, it should be noted that the red dots actually indicate the BRCA mutant samples. This is irrespective of if they're BRCA wild type or mutant. Moving back into cell line assays, looked again to see what's really going on with these PARP inhibitors and this protein. So once again, looked at the COMET assay, and we can see again, if I simply deplete the protein, I get a significant increase in COMET tail length. But interestingly, when I treat with olaparib in the overexpressing cell, they didn't accumulate the same amount of DNA fragmentation. So this could indicate a couple of things. Firstly, the protein could be preventing the DNA damage from occurring in the first place, or it may also be assisting in the repair. So to look at this further, I utilized the panel ovarian cancer cell lines I was using earlier and separated the cells between low expression and high expression. I treated the cells with olaparib and assessed the RAD51 nuclear intensity. So RAD51 is a marker of DNA double-strand breaks. As we can see, following treatment, you get a marked increase in RAD51 intensity, and then in the recovery period, you can see a decrease. However, it was quite evident that in the low expressing cells, they basically had no recovery from the olaparib treatment, whilst in the high expressing cells, they had the peak from treatment, but over recovery time, they were able to actually get back close to the starting point. So once again, suggesting that the protein actually may be required for the repair of the DNA damage. So these low expressing ones are actually going to be sensitive because they will not be able to repair the damage from the PARP inhibitors. It was clear that to progress this biomarker any further into a clinical application, we'd need to assess it within clinical cohorts. We actually found it quite difficult to obtain any publicly available tissue microarrays with PARP inhibitor response data, I guess primarily as the treatment is relatively recent. So we initially simply looked at the expression profile of the biomarker within cancers to identify the staining pattern, as well as things such as the prognostic implication and potential what number of patients we could treat with this PARP inhibitor while using this biomarker. So here I'm just showing our ovarian cancer TMA we undertook, which was 96 cancer samples. So the staining was shown to be both nuclear and cytoplasmic, and with the nuclear intensity actually correlating to patient prognosis. Importantly, it can be seen that 42 out of 96 patients in this TMA was shown to have no detectable signal from the biomarker. So this would basically be the tumours which we would predict to respond quite well to PARP inhibitors. So from the number of TMAs we undertook, we got some quite important findings. So the expression was reduced compared to normal tissue in the majority of cases. But in addition to this, within the tumour environment, a large number had no detectable biomarker, with being 25% in breast cancer, 10 in lung, 40 in ovarian cancer. And this is markedly above that of the BRCA, being only 10 to 15% in ovarian cancer and 5% in breast cancer. So we'd be increasing these cohorts, as well as being able to move PARP inhibitors, which aren't previously really used in lung cancer. As part of this, we actually identified a few issues because we were utilizing a commercially available antibody for the testing. It was shown to be inconsistent between batches, and with the staining being performed at multiple sites, it actually required a lot of optimization for each tumor set. Additionally, if we wanted to progress to a sort of commercial aspect, we would be relying on the company for the antibody, and if production stopped for whatever reason, it really become an unviable biomarker. So to work around this, we began the project with GenScript to develop our own antibody. So this is the basic outline of the progress we were undertaking. So we were able to provide GenScript with 7 milligrams of protein to start it. We decided to actually use full-length protein for our study, just to give the best chance of strong antibodies being raised. It was also noted that GenScript is able to produce the protein and peptides if you weren't able to make them in-house. So after we gave them the protein, they immunized the rabbits, and they would develop the antibodies against the target. Following immunizations, Gents Group performed a number of ELISA tests at different stages, and they provide this information back to us. They also sent the bleeds to us to test in-house. And from these results, we're able to decide which rabbit had the best antibody raised and what we would progress with further. Next, they do the monoclonal hybridization and screening. So from this, the top 10 candidates are sequenced and small batch purification is performed. And then these antibodies are shipped to us for validation selection, as well as the ELISA tests that Gents Group are undertaking. And from this, we're able to select one or even multiple of the antibodies we wish to have large-scale purification. Importantly, we're also able to get the antibody sequence report and even the plasmid produced. So we'll be able to produce the antibody ourselves. And really, the reason we went down this path is to make sure that we weren't required to redo any of the clinical data if we had to get a new antibody into the future. So this is a map where I'm up to in the project so far. So we're awaiting the final antibody, and then we can optimize the new antibody for the biomarker application. And we've established a number of PDX panels as well as retrospective TMA panels that are being lined up for when this antibody is ready. So really what we're proposing, you can see in the summary figure that if tumors are deficient in the biomarker, they would be having impaired DNA repair and they should respond to PARP inhibitors. Whilst the positive tumors would be having a poor response and alternative treatments would be preferred. So this really just leaves me to acknowledge the Cancer Immunity Research Program, as well as my funding, in particular my fellowship from the Advanced Queensland and the funding support from the National Breast Cancer Foundation that have directly supported this project. I'm happy to take any questions.
Thank you, Joshua, for that wonderful presentation. We will now move on to the Q&A portion of this presentation. So please take a few moments now to type your questions into the question box. And while you're typing in your questions, I would like to share a little bit more about GenScript's reagent antibody services. GenScript is an extremely experienced, award-winning custom antibody vendor with almost 50,000 antibodies generated to date. We are able to handle a wide range of project types at different complexity levels, and our monoclonal and polyclonal antibody services are highly customizable and can be tailored to your specific research needs. In addition to proprietary technologies that can help maximize your success at generating a high-quality antibody, we also offer a wide range of specialized services like ELISA kit generation and antibody labeling. Please visit our website to learn more. All right, so let's start our Q&A session. The first question that we have is, Joshua, why do you think your protein or biomarker is downregulated in tumor cells?
Good question. So there's actually a bit of previous data on this protein, and it's really been described previously as a tumor suppressor. And this lower expression has been shown to increase the proliferation rate in some studies, as well as indicating worse patient prognosis. In addition to this, the overexpression of the protein has been shown to increase apoptosis, whilst the depletion has been shown to reduce apoptosis. So I really believe cancer cells are decreasing the expression to avoid the apoptotic pathway and to increase their proliferation.
The next question is, does your biomarker predict sensitivity to any other therapeutic or DNA damaging agents, or is it just specific to PARP inhibitors only?
So I have looked at some additional DNA damaging agents, but none of them have correlated quite as strong as PARP inhibitors. But I've actually also seen the opposite results. So some compounds, such as platinum agents, become less effective in low expressing cells. Now, I believe this to be primarily through the apoptotic roles of the protein, which I just mentioned.
I see. The next question is, why do you think BRCA mutations don't accurately predict sensitivity?
Yeah. So I do think the BRCA is certainly better than no marker, as we can be seen from the clinical data. However, it's quite clear that the wild type BRCA is still presenting with PARP inhibitor sensitivity in some cases. So we were missing this subset if we just used the BRCA, as well as a lot of the BRCA mutant samples aren't responding to treatment. So we can really remove these patients that won't respond.
The next question is, does your biomarker level correlate with the grade of tumor?
So from both our TMA studies as well as the bioinformatic analysis we've done, there's no correlation with tumor grade. with the tumour grades all showing a similar sort of proportion of low or high expression of the biomarker.
And the last question is, what do you think your biomarker protein is doing in DNA repair?
So this is actually a major part of my research, and I'm continuing to work on this. So it's still not quite clear what its direct role is, but I have been able to identify some novel interacting proteins, as well as some co-regulating proteins, which is suggesting which mechanism may be, but I'm still a little way off. So I'm currently teething through this mechanism now. But one of the main functions that's been described to be is a functional structural protein. So we suspect that this protein is actually involved at the site of DNA damage, and it helps orchestrate the repair, and also whilst reporting back to the apoptotic pathways.
And we do have one additional question. Why did you choose rabbit antibodies or rabbit antibody as the species of choice when deciding to generate a monoclonal antibody?
So the main reason I chose rabbit, so certainly in-house what I've been using, a lot of the rabbit antibodies have been better, less background than the mouse ones in my hands. I guess that was a primary choice why I made that decision. But yeah, I think that was the main.
Awesome. Okay, unfortunately, we are out of time and have to conclude this webinar. Once again, responses to any questions that we were unable to answer during this webinar will be emailed to you directly. Thank you, Joshua, for being so generous and sharing your research with us. And thank you to all who have taken the time out of your day to join us for this time of learning. Have a great week ahead.